Hungry fat cells can deprive cancer of ingredients it's nutritious. Scientists have found a way to starve cells cancer with the help of brown cells of fat tissue. These voracious cells quickly use nutrients, depriving cancer cells the fuel they need to grow and survive. Using this method, researchers beat 5 different types of cancer in lab experiments. Scientists inspired by the methods used in liposuction and plastic surgery have developed a technique that can deprive cancer cells of nutrients. First, they transformed the ordinary fat cells in brown adipose tissue cells that aggressively burn calories to create heat. Then they implanted the modified cells close to tumors developing in mouse models, as well as plastic surgeons carry fat from one part of the body to another for improvement of appearance. As a result of this procedure, the modified cells quickly used up available nutrients, leaving nothing to cancer cells. Results and description of the research appeared in the journal Nature Biotechnology. Brown adipose tissue, also known as brown or beige fat, is one of the 2 main types of fat found in people and other mammals. The color of this tissue is due to a large number of mitochondria. Initially, it was believed that brown adipocytes, or cells that build tissue fat, they are only found in infants and hibernating mammals, but in recent years, it has been discovered that adults also have brown fat. His main function is to produce heat by burning calories as opposed to white fat, also called yellow, whose main function is storage of energy. Researchers from the University of California, San Francisco, UCSF, they used brown fat cells to deprive cells of the cancerous nutrients. Using a gene editing tool, CRISPR-Cas9 converts white adipose tissue cells into cells brown fat that is known for its ability to burn calories to generate heat. Then they put them in contact with the mouse models of several cancers. Modified fat cells absorbed quickly key nutrients, effectively depriving cancer cells of fuel. Interestingly, this approach worked even when modified cells' fats are placed relatively far from the tumor. Important in these studies is that the developed method is based on existing medical procedures, which can lead to faster transition from clinical trials to use in patients suffering from cancer. We routinely remove fat cells for help with liposuction and we restore them with plastic surgery, he said. Nadav Ahitov from UCSF, co-author of the publication. These fat cells can be easily manipulated in the lab and safely put them back into the body, which makes it an attractive platform for cell therapy, including cancer treatment, he added. Authors of the publication have previously pointed out the research, which have shown that cold exposure can inhibit the development of cancer in mice. They also knew that cold activates brown fat cells that they use nutrients to produce heat and heat the body. They acknowledged that inhibiting the development of cancer in mice after exposure to cold results from the fact that cancer cells are starving because brown adipose tissue has worn out all available resources to heat the mice a bit. But exposure of oncological patients in the cold may not be the best option, so scientists have begun to develop strategies to use the brown fat. They assumed that they could design it so that it consumes all available nutrients without the need to activate it in the cold. They used the CRISPR-Cas9 technique to activate genes that are dormant in white fat cells but are active in brown fat cells, in the hope of finding those that will transform white fat cells in the ever larger brown fat cells. In this way they found a gene called UCP1. In further studies, they bred transformed from white fat cells brown fat cells together with cells with cancerous Fat and cancer cells were in separate spaces but had access to a common part where researchers placed the nutrients. It turned out that cancer cells do not have the smallest chances in the food race with brown fat. In our first experiment, few cancer cells, it survived. We thought we broke something. We were sure it was a mistake. So we have repeated the experiment many times. We watched this every time the affected cell, admitted HkUzY. Experiments on petri dishes. Lignite fatty tissues effectively deprived nutrients of two different types breast cancer cells, as well as colon, pancreatic and prostate cancer cells. But it was still unclear how brown fat would cope in more realistic conditions. In the next step, researchers used cell clusters fats grown in the lab to see if they can beat cells cancer when implanted next to tumors in mice. In this case, hungry brown fat cells have also won the race for nutrients. The approach this effectively inhibited the development of breast, pancreatic and prostate cancer cells. Implanted cells of beige fat were so good that inhibited the development of pancreatic and breast tumors in mice that were genetically predisposed to the development of breast cancers. Moreover, the lignite cells' fatty tissue consumed nutrients faster even when they were left implanted relatively far from breast cancer cells. Scientists have also tested brown fat on tissue. The human Jennifer Rosenbluth, also from UCSF, provided samples of mastectomy containing both fat and cancer cells. Scientists they converted the fat cells from a sample taken from one patient and they were tested by the cells of breast cancer of the same person. In tests on the dish, petri brown fat starved cancer cells. The same thing happened when the researchers implanted both types of cells together into the mouse body. Different cancers prefer different nutrients. To be an example of such, pancreatic cancer, when glucose is lacked, is taken from uridine. Scientists have modified fat to consume only specific ingredients HkUzY, in this case only uridine. Medieval fat cells prepared in this way, they easily won the food race. This suggests that fat can be adjusted to dietary preferences of cancer. Fat cells can be easily obtained from patients. It's good, and that's good. They grow in the laboratory and can be modified to express different genes, and they took different biological roles. What's more, they don't cause problems when will be reintroduced into the body, without moving away from the place in which they have been implanted and working well with the immune system. Researchers believe that fat cells can also be programming to emit signals or perform more complicated tasks. We believe that these cells can also be designed to they detected glucose in the blood and released insulin in diabetes, or absorbed iron in diseases where there is excess iron, such as hemochromatosis, admitted HkUzY.